This video introduces you to an important class of chromatin proteins, the nucleosome remodelers. So far, we've talked a lot about chromatin structure and about nucleosomes being part of the basic packaging. We've discussed how the chromatin can be opened up or closed, thus impacting transcription by altering DNA and histone modifications. But what if that is not enough? What if we actually need to move some nucleosomes around to make a specific DNA sequence, like a promoter, accessible to the transcription machinery? Changing access is something that has to happen a lot in a cell. For example, in response to an environmental stimulus like heat or cold, or to a hormone signal. This DNase I hypersensitivity assay reveals how nucleosome positioning is altered in response to hormone signaling at the tyrosine aminotransferase gene in rat. The enzyme DNase I can cut regions of open chromatin, often between nucleosome. It lets us see open regions of chromatin. If it doesn't cut, we get large pieces of DNA on the gel shown above. If it cuts a lot, we see small pieces. In this assay, where there is no hormone on the left side of the gel, most of the signal we see is at the very top of the gel. If we treat the cells with hormone, called induction here, we see new, smaller bands appear, which means a new site in the chromatin of this gene has opened up. If we wash the hormone off the cells and wait a little while, this band disappears and we go back to the original uninduced pattern, illustrating that there are mechanisms to move around the nucleosomes. The proteins responsible for moving around the nucleosomes in the previous experiment are called nucleosome remodelers. They use ATP to carry out their work, and they can have several different functions. As shown in A here, they can slide nucleosomes, evict nucleosomes, or facilitate histone exchange. As shown in B, they can also assist in nucleosome assembly, and lastly, shown in C, they can affect nucleosome spacing. All of these processes can have important impacts on access for transcription machinery. There are four main classes of ATP-dependent nucleosome remodelers. They all utilize ATP, but their exact functions differ and can include one or more of the ones we've discussed in the last slide. The four classes are the switch-SNF subfamily, the ISY subfamily, the CHD subfamily, and the INO80 subfamily. Given the various different activities nucleosome remodelers can have, it is no surprise that they participate in a variety of protein complexes. This is illustrated here for the ATPase ISY. ISY participates in at least 6 different chromatin remodeling complexes, each with slightly different function and potentially different genomic targets. Here is a model for how nucleosome remodelers might carry out their function. In this case, sequence-specific transcription factors recruit a histone acetyltransferase like SAGA. This histone acetyltransferase produces histone acetylation, and the bromodomain proteins within the remodeling complexes can now interact with the acetylation and bring down the nucleosome remodeler. Now, nucleosome remodeling can take place. The histone acetyltransferase now acetylates a non-histone target in the remodeling complex, which competes for the bromodomain reader domain for binding. As the bromodomain protein binds to the remodeling complex rather than the histone, the remodeler disassociates from the DNA. In summary, nucleosome organization is not static but very dynamic. Nucleosome remodelers are specialized protein complexes that can locally change nucleosome organization. Nucleosome remodelers can slide nucleosome, evict nucleosomes, or exchange nucleosomes. Nucleosome remodelers are large, diverse protein complexes. They interact with histone-modifying enzymes to control chromatin structure.